Biotechnology is a research-oriented field. So you need to possess some technical skills along with soft and transferable skills in order to get placed in a life science industry or a biotech or biopharmaceutical company. Hello everyone. Today's topic is top 15 research skills every biotech or life science candidate must have. Although it goes without saying, technical skills hold an added value for the aspirants. If someone has to work in the most decent and repaying job posts, they should be acquainted with the requisite R&D technical skills. Furthermore, they also should be aware of the latest technological advancements, which are the following I'm going to discuss with you. The first technique is protein purification. Protein engineering techniques are an essential part of customizing or producing proteins with specific properties which can be applied in various industrial processes. So thus, needless to say, they are very crucial to biotechnological research. However, these methods depend heavily upon being able to isolate and purify the desired proteins so that their physical and chemical properties can be understood along with their tertiary structures and interactions with ligands and substrates. So the intensity to which this purification process is pursued depends upon the use to which the protein is to be put. For instance, pharmaceutical and food proteins need to be brought to a high grade of purity and pass through several sequential steps, as few as possible, since at each step some protein will inevitably get lost. So which all techniques you can study under that? The first technique to learn is gel electrophoresis. For any molecular analysis such as analyzing DNA, RNA and protein, they have to be done by gel electrophoresis, which is nothing but the separation based on their size by applying an electric field as the DNA is run through agarose gel. So it is very important to learn using the electrophoretic apparatus and also to cast the gel properly. The quality of gel will provide a transparent and accurate result. While preparing for casting, the most important part is an accurate proportion of the ingredient, that is agarose percentage, polyacrylamide percentage, as this will affect the pore size, which in turn will determine the experimental outcome. So these are the technical details which you need to learn as a life science expert. While agarose gel is used for the analysis of DNA, PAGE, polyacrylamide gel electrophoresis, and SDS, sodium dodecyl sulfate PAGE, they are used in proteins. Agarose gel electrophoresis is the most effective way of DNA analysis based on size. This is widely used in molecular biology and biochemistry labs in areas such as forensic science, conservation biology, and medicine. So another technique which also goes with this topic is isoelectric focusing. So what is isoelectric focusing used for? It is the most commonly used technique for the separation of proteins. IEF separation, isoelectric focusing separations, they are based on the pH dependence of the electrophoretic mobilities of the protein molecules. Now, during the separation process, the sample accumulates in specific and predictable locations in the medium, regardless of their initial distribution. This focusing also distinguishes the IEF method from various other modes of electrophoresis. So it's actually a relatively low cost and very routinely used laboratory technique. This has a strong worldwide user base that supports a commercial pipeline of instrumentation as well as consumable products, and thus will certainly remain a very demanding process if you are going to apply for any research work. Some key applications of the techniques. So though this lab is taught at graduation level, itself to all life science students, it's very limited. So one must thorough the entire gel electrophoresis protocols, including process, its limitations, error rate, applications. So this technique once masters will be with you until you are into your research field. So as you can see, all the important things that you come along most of the time in your research work. So most of these will definitely have, this is the least thing that you are expected to know, that is gel electrophoresis. The second technique to know is macromolecule blotting and probing. So what is it? Blotting is a common lab technique widely used in the field of cell and molecular biology. It is an extremely useful multi-step technique used in the identification of different biomolecules such as DNA, RNA, protein during different stages of gene expression, and it is also used for identifying specific proteins from a complex mixture of proteins after extraction from the cell. So the applications are actually a lot, right? They are used to transfer DNA, RNA into a blotting membrane, often after gel electrophoresis, so that they can be stained or radioactively labeled, and then they are visualized. So this is a looking at its wide range of application. It is a must learn lab technique for all who want to build a career in research. Now only theoretical knowledge is not going to suffice. Sufficient hands-on training experience with all types of blotting techniques will help you conduct experiment and research at much ease in your PhD or work. So what are the types of different blotting? Southern blotting, which is used in gene mapping, gene discovery, transgenic forensic. Northern blotting, it is for the mRNA level, gene expression analysis. Western blotting, it is for the protein, so identification of specific antibody in complex or any confirmatory testing for some diseases. And other alternative blotting techniques are also available based on the experiment requirement. So one can know all the principles, underlying principles, and especially the techniques, how it is done step by step, what are the tips and tricks to get the technique correct, to get accurate results. These are the things which one should learn properly. Next concept, next technique you need to know is PCR. Yes, PCR, this is one of the most important techniques used in molecular biology, and it is basically used to copy DNA. It allows a single DNA sequence to be amplified into millions of DNA molecules. So what can PCR be used to? Introduce mutations within the DNA or introduce special restriction enzyme sites. In addition, PCR is used to determine whether a certain DNA fragment exists in a cDNA library. So it is an extremely crucial method in molecular biology. It is not just a groundbreaking technology, but actually it is also very economical. By thousands of duplicates of DNA could be made using just a single DNA strand for a base. The polymerase chain reaction method is extensively used in medical and biochemical lab. It is an essential component of nearly all kind of tests. So different PCR you can types of PCR you can learn, but make sure you know this reverse transcription RT-PCR for amplification of RNA and quantitative PCR to measure the amount of RNA or DNA present. Applications are many listed here. They are only a small portion of other. If you have a desire to make an impact in the world of research using this method, it can be of a great help to you. The most advanced PCR devices are being developed by various manufacturers worldwide that you should know about these machines as well. Keep up to date your knowledge of the latest developments and also in the field of PCR machine techniques and use by reading research papers and trends in the research. Next technique to learn is molecular gene cloning and expression cloning. So it is simply, which is called as gene cloning. It is a molecular biology technique that is used to assemble recombinant DNA molecules and to direct the replication within the host organisms. It's a basic technique in molecular biology and biotechnology laboratories, and it's a very useful tool to study a gene, modify the gene, reintroduce the modified gene into some neutral host or another host, or even to produce protein. You can perform molecular cloning by incorporating a piece of DNA into a plasmid to make more identical recombinant DNA in a living host. So this technique helps scientists understand the protein function. The DNA that codes for a particular protein is cloned or copied using PCR into an expression vector which is called as a plasmid. So you must know the processes of plasmid isolation, which you might have to do on a regular basis. The plasmid is introduced to either an animal cell or bacterial cell, so you should also know expression vector constructions. Then what all promoter elements will be there in the plasmid which can stimulate high expression of the desired protein, so that the enzymatic activity can then be examined. So other than that, you have different types of cloning types which you can learn the protocol about. Next technique is UV visible spectrophotometry. Yes, they are mostly used for both qualitative and quantitative studies of biomolecules such as proteins or nucleic acids as well. So UV visible spectroscopy, it is used in molecular biology as well as in microbial studies, as it is possible to assess the quantification, purity of proteins and nucleic acids, as well as determining the microbial growth curve. And similarly, there is one more technique along with that, that is mass spectroscopy. It is used for the determination of molecular weight of proteins. Then you have NMR also. It is used for structural refinement, prediction of protein structure, to understand the mechanism of the secondary structure of a protein by protein folding, conformational dynamics, DNA RNA stimulation, and protein stimulation. So ensure that you at least know how these are done. CD spectra, IR, atomic absorption spectrophotometer, and definitely, as I said, along with that, you should also know mass spectrophotometry. So it is one of the analytical technique that ionizes chemical species and provides and sorts the ions based on their mass to charge ratio. So it has become a very essential analytical tool for biological and biomedical research. It provides a rapid and sensitive qualitative and quantitative analysis of biomolecules, that is, your peptides, proteins, oligosaccharide, lipids, DNA, RNA, and along with that, drugs and small molecule compounds and even metabolites. So, in combination with a variety of modern separation technologies, MS has been successfully applied to modern biology and biomedical research. So, this is used applied in high throughput proteome wide analysis of proteins, protein protein interaction, protein post translational modifications in cells, tissues, or organs. And besides, it can also be used in the production of novel medicinal agents as well. So ensure that this is a must learn technique for life science students. Another important technique is next gen sequencing. So earlier, what we used to do, like using capillary electrophoresis based Sanger sequencing, the Human Genome Project took over 10 years and cost nearly $3 billion. Now, next gen sequencing, in contrast, it makes large scale whole genome sequencing accessible and practical for the average researcher. If it enables the scientists to analyze the entire genome in a single sequencing experiment or sequence thousands to tens of thousands of genomes in 1 year. So it quantifies the discrete digital sequencing read counts, offering a much broader dynamic range. Researchers, and it enables scientists to analyze entire human genome in very less time. You can learn about NGS based RNA sequencing, Illumina next gen sequencing. So researchers in different fields utilize this to make breakthrough discoveries. For example, to study the genetics of COVID-19 susceptibility, compare the genomes of the patients to help uncover genetic factors associated with susceptibility. Or if scientists want to explore the tumor microenvironment, elucidate their gene expression patterns, gain insights into drug resistance and metastasis. And another one is cell-free RNA sequencing, for if any scientists want to go for biomarker discovery and non-invasive health monitoring. So in case these are the research areas you're interested in, then definitely this is a must-learn technique for you. The next in line is chromatographic techniques. Nothing to say about this. It's the most common analytical technique used for separating and analyzing a mixture of chemical substances, and it is a vital part of almost any protein purification strategy. The numerous and different chromatographic techniques are mainly used for the production and analysis of proteins. This can be classified as per the physical principle that is involved in the process of separation. that is thin layer chromatography, affinity, size exclusion, ion exchange, HPLC. So it plays crucial role in various types of industries like pharmaceutical, food or chemical industry, wherever you talk about. Ensure that you know how these processes are done and how you are supposed to get the accurate results, what all equipments are used and how do you handle those equipments. Okay, everything is very important. Next are bioinformatics tool. Yes, this is not a technique to learn as such, but yes, you have to know about what all tools are then and how to operate those tools. So if you are a candidate who is interested in becoming successful in their research career, you should have a thorough understanding of bioinformatics tools these days because it's a necessity. In addition to being effective in wet lab methods, researchers working in the area of genomics and proteomics, they require a complete understanding of soft lab techniques also. So this field is an emerging interdisciplinary research field that is becoming increasingly used in all kinds of research, be it cancer research or mapping the human genome. So start learning bioinformatics tools and techniques at a very early stage of preparation. So to compare the whatever tools you have to master, to compare the homology of sequences, you should know BLAST, or to identify the structure of proteins, you must know RASMOL, or to create the primers, then NEB cutters. So other tools along with that need to be mastered, right? So because the vast majority of proteomic and genomic research needs up-to-date information, scientists should be in routine practice of accessing databases of biotechnology and molecular research to find any information that has been updated. Bioinformatics in general is a broad field with numerous methods such as molecular docking, molecular modeling, and even programming languages such as Python, C, C++, SQL, and all the down goes the list. Another topic which comes along with that is statistical analysis tools. Yes, in the medical research field, stretching from systematic reviews, meta-analysis, clinical trials, exactness, precision, these are very paramount informations which you have to collate together. So there are vast universe of statistical tools which are used in biological research by scientists, physicians, and industry R&D professionals. Like for example, Stata. Lot of statistical features are present here, from descriptive analysis, cross tabulation analysis, to even more advanced techniques like structural equation modeling, probability models, survival analysis, time series, multi-level models. So they will allow users to have control over data, variables, and also statistical compilation of the groups. R, it commands a strong fan base, which is contributed by data scientists, bioinformatics, and medical researchers. It covers and expands of disciplines from cancer research, clinical analysis, molecular biology, phylogeny, to even meta-analysis. GraphPad Prism is a popular among biologists in academia and also in research industry. It also comes with functionalities that allows researchers perform laboratory research and clinical trial test using the t-test, one-way ANOVA, contingency table, probability models like logistic regression model and all that. MATLAB, yes, it is gaining popularity these days among scientists in the area of engineering, numerical analysis, linear algebra, and even in image processing. Next tool in line is flow cytometry. This is a beneficial technique for cell sorting, that is, separating different types of cells from a homogeneous mixture of cells. So this is a laser or impedance based technique which is used in cell counting, cell sorting, biomarker detection, protein engineering by suspending the cells in a stream of fluid and passing them through an electronic detection apparatus. This instrument is so sophisticated that it can be used to identify multiple physical characteristics of a single cell. This entire principle depends on the light scattering properties of the cell of your interest, which is tagged with either dye or monoclonal antibodies on the cell surface or some intracellular specific molecules. So this FACS allows the study of multiple populations of cells within a limited time. This instrument is highly essential for cell biology and animal or plant tissue culture laboratories. Thus, candidates who are seeking a research career in these areas, they must get a hands-on experience on this technique as soon as possible. Next important technique is microscopy. I don't have to mention about it that it is used extensively in a vast area of science topics, subjects, especially to comprehend the fundamentals of life and carry out vital research regarding it. Right? So, a particular field in science can't make sense without a microscope. For example, the histology field. It's extremely fascinating to study tissues at the microscopic scale. It will assist us to learn the functional morphology or dynamics of tissues. So this is why this particular thing is growing in popularity to learn. Confocal microscopy, electron, fluorescence. Be sure you know how to operate this properly and get the proper images, right? A particular field in science can't make sense with microscope. And real-time microscopes, they will record in this modern technology that is used to build microscope. The most intriguing are ones that operate in real time. That means they provide a video footage of the interactions between organisms and the intracellular dynamics. So they record really exciting events. That update also you can learn about. Also important are immunotechniques. So they are wide variety of diagnostic techniques which are used to measure and characterize the specific immune responses. Immunotechniques also help us to perceive about the antigen and antibody interactions which are taking place and identify particular protein in biological sample. So they allow the physicians to refine the immune system through genetic manipulations also. Immense variety of techniques are there for separating and characterizing various molecules. You can learn about all these techniques: ELISA, RIA, rocket immunoelectrophoresis, counter current immunophoresis techniques, and immunohistochemistry. So they are routinely used for the diagnosis of infectious agents such as viruses and other substances in blood. So if you're going for a pathology job, then definitely immunotechniques are something which you must learn. Another important set of techniques are cell-based assays. Yes, they can be used in both biomedical research and drug discovery screening applications to efficiently quantify cytotoxicity, biological activity, biochemical mechanisms, and off-target interactions. The advantages of cell-based assays include the facilitation and generation of complex and biologically relevant data. Unlike traditional biochemical assays, cell-based assays are more physiologically relevant and can assess compound characteristics simultaneously. Cell viability, cell proliferation, these assays can viability they can be used to evaluate the cell health, and they are often assessed using some dyes such as trypsin blue, calcine aim, or with fluorescent readouts. Cytotoxicity is there, cell senescence, death. You must know how to do all this process. Cell migration and invasion, they can be evaluated in vitro with widely used membrane-based migration system like the Boyden chamber assay, and the kits are often used in cancer research to study cell motility and invasiveness in response to chemoattractant gradients. Second last technique to learn is centrifugation. Most of you must be already knowing this. Broad range of applications in both the labor and the industrial sectors. And not just this is the procedure to isolate to miscible substances, so that as well to study macromolecules or hydrodynamic property, but this is also a significant and widely used method of research in biochemistry, molecular biology, and cell biology. In the food and chemical industry, special centrifuges handle a constant stream of particle-laden liquid. So alongside these instrument and lab methods, some basic laboratory procedures also must be learned. These techniques are very simple and you are expected to know them, like autoclaving, sterilization, cleaning the equipments, as well as common preparation of reagents, and like this the use of centrifuge, even how to use laminar airflow. Then also preparation of media plating, culture of microbes, that's going to be the last important technique. And they also include serial diluting, optimizing different equipment and water baths, creating cotton plugs for culture tubes. So these are very minimal amount of knowledge that you're expected to know. So last in the list is the tissue culture techniques. Yes, it is one of the most widely used technique in any biological or biomedical research lab. So if you are looking for a placement or job, tissue culture will be a very valuable technology skill to have. Okay, so it involves the growth of cells separated from tissues or multicellular organisms, or tissues from separated from organisms, by using a liquid, semi-solid or solid growth media such as broth or agar. It typically refers to the growth of animal cells and tissues, whereas the term plant tissue culture is used for the plants. So it is an in vitro technique which is important for the study of biology of cells from multicellular organisms. It's an ideal in vitro model of the tissue in a well-defined environment that can be easily manipulated and analyzed. So along with that, even cell culture techniques also come into picture, especially the microbial cultivation. It is one of the most effective methods to investigate the physiology, biochemistry, genes of microbes along with the screening of antibiotics. So it is essential for microbial, cellular as well as molecular research, biological research. It can be used to study mutagenesis and carcinogenesis by the application of potent mutations. The cells can also be screened for drug discovery and development, and they can be also used for the production of essential metabolites that can be used for production of items on a vast scale. So there are learning this technique is one of the essential requirement of cell biology laboratories, like maintaining a sterilized environment, using suitable media, incubation, storage of cells, subculturing at appropriate times. It is crucial for cell culture. These set of techniques, they include the knowledge of how to design and the equipment used in the lab, safety aspects of cell culture, sources of the cell line, how to source them, cell types and the culture characteristics, also how to store the cells through cryopreservation. The cell culture protocols also you should be aware of, and authentication of cell lines is also a must know technique. A special mention goes out to CRISPR technology. Yes. So although these are standard lab techniques which we discussed, the researchers must be aware that there are these important techniques like this upcoming ones such as CRISPR, and knowledge about them is definitely a must have in the future. You would be already aware that Biotecnika is conducting genome editing with CRISPR virtual internship, and you will get to know to learn the basics and advanced concepts of CRISPR genome editing. And these methods are typically learned after an experience in the laboratory. But however, if you want to launch an academic career, a scientist must be technologically competent in every aspect. So along with that, you can also go for this. So who is this beneficial for? For BSc and BTech students from biotechnology, microbiology, genetics, biochemistry and other biosciences, MSc and MTech candidates who are looking forward to learning this technology in depth, and also research scholars and PhD aspirants who should gain experience and knowledge on advanced tools used in research. I would like to conclude the session by informing you about all the techniques. Different certification courses are available at Biotecnika. So any of the techniques you want to learn, all you have to go is in the stores, Biotecnika stores, and at least you can learn all that you need to know for any techniques that you would like to learn about, right? Thank you, everyone. I'm pretty sure this session is going to help you in your career ahead, and please start following up and learning these courses one by one.